Hey everyone, this is UTC Tips and Tricks. Today I'm going to show you a quick overview on how to use various bioinformatics tools. Before we start, sorry for the weird recording audio, I don't know how to record. So first, you'll be provided with an unknown DNA text file. Keep in mind that this is pulled off the internet for demonstration purposes only, so don't commit any academic offenses. You're going to want to copy paste this, we'll be using this a lot. First thing you want to do is go to BLAST, which is a DNA identification website by NIH. Since this FASTA sequence consists of DNA nucleotides, we're going to want to go to the nucleotide BLAST sequence. So copy paste your FASTA sequence, do not touch anything else here, click BLAST, and this might take a few minutes, a few seconds. It really depends on how many people are using this. Oh, this is too small. And so you can see here that there are a lot of results. In this percent identity column, we're aiming for like the highest percentage. If I'm feeling nice, I'll provide you with a Google Doc or something like that in the description or comments. That'll tell you how to analyze this further. But you'll have to find peer-reviewed articles anyways to explain your findings, and unfortunately this isn't peer-reviewed. I could be giving you false information. Anyways, there are lots of 100% here, so we're going to narrow that down later in the tutorial. The FASTA sequence provided by your TA will be longer, which means you'll have a more narrow search. So what we're going to do is we're going to click this first one, and we get to this page. In your report, you may want to note what these mean, for example, the gaps, strands, identities. You also want to keep this tab open. So in your report, you may want to screenshot this, like this alignment section, and the descriptions, specifically like the top 100% ones, or whatever can fit on your page. So yeah, we'll keep this page open. Next thing we'll do is go to NEBcutter, which is simulating DNA interaction with restriction enzymes and gel electrophoresis. So again, we'll paste the FASTA sequence, press submit, and these are, I believe, all the restriction enzymes that can be cut through this sequence. I'm not too sure. If you want to see specific restriction enzyme sequences, click the ones you want over here in custom digest. Keep in mind that not all restriction enzymes may be able to cut through. For example, see look, there's no ECRI. Let's just choose random ones here. So once you have the ones you want, you can see over here, click digest, and here are your custom enzymes in action. Now we'll go to gel electrophoresis, and really what you can do is play around with these drop-down menus. As you can see here, it's not really the best, the most ideal to visualize. So if we go to drop-down menus, you can see that it's a bit easier to see the spaces in between and everything. It's just visually nicer. Something to also note is the CpG methylations and just generally methylations here. If you haven't already, you'll learn about them in your second year cell bio courses. So if you want a screenshot for your assignment, this is one screenshot, this is one screenshot, and this is another screenshot. On to the next website, ORF, which is Open Reading Frame. This finds the sequences of DNA that can be turned into protein. So again, copy paste the FASTA sequence, don't touch anything else, press submit, and here it is. In my assignment, I just screenshot this whole thing just in case. We'll be using this protein sequence a lot, so don't delete this website. Next, we'll go to the sequence manipulation suite site, whatever. We'll use this site for the protein, where is it? I can't even find it. Protein isoelectric point and protein molecular weight. Here. Okay. Paste the protein sequence this time. Where is it? Here. paste the protein sequence this time. Press submit, and you can see that the pH is 4.63 here. You can do the same with protein molecular weight. There. So that's 18.36 kDa. You don't need to you don't need to screenshot both of these numbers, but you do need to have it in your assignment, I believe. Okay, so yeah, note down the numbers. Our second last website is PSORTb. These are random names, which helps with protein localization, aka where the protein is in the bacteria. So for this one, paste everything related to this protein. So even this ORF1 thing at the top. Do that super quickly. Okay, so results via web. And for gram stain for bacteria, it's usually gram negative, but just in case, I think you need to Google the species name and just say like, oh, is this gram positive, gram negative, stuff like that. So let's go back to our first result. Let's do, is this gram positive or gram negative? Gram negative, amazing. So, put negative here via the web, submit. Okay, so here are the results for localization. If you go to this bottom area here and look at the highest number, in this case it's extracellular, meaning that the location over here is where your bacteria is most likely to be found. So keep in mind that when we go to our 3D modeling, which we'll do now, just note, for example, the secondary location, which is flagellar, whatever it is. Screenshot this whole area and save it for later. Why is this? Okay, anyways. So this is probably the step where the most important interpretation is required and where it's most open-ended. So check with your TA if you're doing this properly. I didn't do it right the first time. Make sure you do. I want to be as specific as I can when looking for my protein. So let's go back here. Okay, I don't know how I did that. So pasting this whole thing, I'm pretty sure it won't give us any results. So we got to be less specific, but still enough so that we can identify our protein. So let's say we can, what is it? Copy paste this subspecies, and along with the protein that's most likely in, which is like flagella, right? So we're probably thinking that this protein is, yeah, again, outside of the bacteria. Okay, yeah, again, there will be a lot of results. Yeah, there are a lot of results, and again, I pulled whatever this sequence is off the internet, but yours will be a bit more straightforward. For example, over here, when you search up your DNA sequence, it might also come with a gene, maybe like FLIC or FLIJ, whatever it says. It might be ideal to match the gene, whatever it says on this title. So in this case, it doesn't say it doesn't say what the gene is, but for my assignment, it did, which helped me identify the correct protein. So yeah, again, it's not going to be chaperone because we're looking for flagellar. This is a break, so it must be something like this. So I'll click that. And this is a wonky kind of flagellar. I don't think, I'm not sure if this might be right, but again, you need to check with TA and everything. It does kind of look like one because it is long, I guess, but yeah, this is what your end result should look like if you do have a similar structure. And you can see here for your assignment, you can talk about which parts of the protein are very high in confidence, which are very low, and yeah, I think that's really it. Yeah, so like if you can see FLIS, for your assignment, there should be FLIS somewhere in this title. Yippee! I think we're done. This is what I did for my assignment, and I got near perfect. I think if you do the same things along with confirming with your TA, you'll be good to go. So yeah, good luck, and hopefully this video helped.